Hello again. All right, as I mentioned at the end of the last lecture, we're going to head over to the pancreas and see what's going on in the pancreas to help us out with digestion. All right, so way back at the beginning of the course, we learned how the pancreas has both endocrine and exocrine functions. And endocrine functions, again, refers glands produce substances that get secreted, released to the outside. Endocrine glands produce substances that enter the bloodstream and travel throughout the body. Exocrine glands produce substances that get released onto some body surface, a mucous membrane inside the body or the skin on the outside of the body. So the pancreas has both endocrine and exocrine functions. The endocrine functions we learned way back in Unit 1 when we were studying the pancreatic islets, or the islets of Langerhans, that make insulin and glucagon hormones that help us with controlling our blood sugar levels. The bulk of the pancreas by far, though, consists of cells that are involved in exocrine functions, and it's going to produce a substance called pancreatic juice, which has lots of enzymes in it, and it's also an alkaline fluid, which means it has a high a pH above the neutral range. And if you think about it, that makes sense, because what's coming in from the stomach, the chyme coming in from the stomach, has a low pH. The vast majority of the parts of our body have a pH of 7.35 to 7.45. So as that chyme is coming into the duodenum, we need to zap it with some alkaline fluids to help bring the pH back down into the normal range. Because the duodenum, the intestinal tract, is not an acidic environment. Some people have that misconception. The stomach lining is an acidic environment, but once you hit the intestinal tract, you go back to a more normal pH level for digestion. So there are cells in there, or clusters of cells, called acini or acini, which is an Italian word, and they secrete pancreatic juice. The cells can also be called acinar cells, or acinar cells. ACINAR is another name for those. So they produce pancreatic juices that travel through little passageways, little ducts, that eventually lead to the large pancreatic duct, which is going to take these juices over to the hepatopancreatic ampulla that we were taking a look at a few minutes on the last lecture clip when we were discussing the liver, and then those get released into the duodenum. Then we're going to see these acini cells, or acinar cells is another name for those. They are granulated. If you look inside them under a microscope, they've got lots of little membrane-covered granules inside them. Kind of like the granulocytes when we were learning the white blood cells, kind of the same concept. Those are storage compartments for what are called zymogens. Zyme, like enzyme, and ogen, you've seen that several times now in Biology 202. Those are inactive forms, usually of some kind of enzyme, like we had with angiotensinogen, for example, or fibrinogen when we were studying blood clotting. Now we have zymogen. And so these are inactive forms of digestive enzymes that have to be altered a little bit before they're going to work. So those can also be considered pro-enzymes. Pro, that prefix means before they become truly active enzymes that have digestive functions. Those are stored inside these cells. All right, so as I mentioned, the pancreas is full of these little subunits called acini, and acini are kind of spherical in shape, and this is one acinus, or acinus, which is the singular form. And so it's composed of a whole little cluster of these acinar cells. And the little spots you see in there, those are the zymogen granules that contain these inactive enzymes, which when they become activated will help us digest our foods over in the duodenum. So these substances get secreted into this space or lumen here, and then that leads to ducts little tubes or passageways that are going to carry those fluids. The duct cells, as we'll see, those cells that actually line these compartments, produce an alkaline fluid. Okay, so we make sure the pH is high in this pancreatic fluid, again, because that's going to help us neutralize or convert the acidic chyme coming in from the stomach back to a more normal pH level in the duodenum. All right, so the pancreas makes lots of these pancreatic juices, 1,200 to 1,500 milliliters per day. That's 1.2 to 1.5 liters. So that's a lot. You don't think about your pancreas sitting there behind the stomach making that much fluid. It is a very watery fluid, alkaline pH. So the pH is 8. That's on the alkaline side. And again, when you mix this with the chyme coming in from the stomach, that's going to bring the pH back up to a more normal level for our body. And if you remember when we were talking about the stomach, the chyme just comes in little bursts. So it's not like you don't get a big gush of acidic chyme coming in from the stomach. The reason that's important too, the tissues of the duodenum and the rest of the small intestine would be damaged by the low pH of the chyme in the stomach. They don't have remember, the stomach lining has a mucus barrier that actually protects it from the low pH of the fluids that the stomach itself is manufacturing. You don't have that feature, that protective feature, once you get down into the small intestine. There are lots of electrolytes in pancreatic juice, but mainly our friend HCO3 with a negative charge. Remember, that's bicarbonate. And bicarbonate ions raise pH. Those are alkaline molecules or base molecules that help bring pH back up. Lots of enzymes in pancreatic juices, so we're going to start talking quite a bit about enzymes that are used for digestion as we go through the rest of the info on the small intestine. Amylases, mentioned those with saliva before, those digest starches. Lipases digest lipids. Nucleases digest nucleic acids, DNA and RNA, which we do have in the foods because we're eating, when we eat foods, most of the things that we're eating are, or lots of the things we're eating are cells from plant and animal tissue, so there are DNA and RNA in there that have to be broken down. And then lots of proteases as well. Proteases are enzymes that digest proteins. All right, so these proteases, you have to be careful with these because you don't want them activated until they really need to be activated and in the right location to start breaking down your foods, because cells throughout the body, cell membranes of any living thing, this is one of our cells, the membrane always has in its surface lots of membrane proteins, things like receptor proteins, for example, that have important jobs. So you don't want proteases in an active form just floating around in your body fluids where they could attack the proteins in your own cell membranes, or proteins that are floating around in your body fluids that have important jobs they're supposed to be doing. So you have to make sure you only activate these proteases when needed. Okay, so the acinar cells produce a proenzyme called trypsinogen. That's the inactive form. It gets activated to trypsin. Trypsin is an active protease, and that occurs once the trypsinogen arrives in the duodenum as part of the pancreatic juice. When it gets squirted in there, the cells that line the duodenum have an enzyme along the brush border called enteropeptidase. So if you guys remember when we first looked at the inner lining of the small intestine, we said that the cells, they're actually more of a columnar shape. They have lots of little waves and folds along the surface that faces the lumen of the small intestine, and that increases surface area for absorption. And there are lots of enzymes sitting in the membrane along those microvilli. That's what those little folds are called, That's the microvilli or the brush border. So there are enzymes there called enteropeptidase enzymes that take trypsinogen and convert it to trypsin. There's another protease called procarboxypeptidase, and then there's yet another one called chymotrypsinogen, and these are activated by trypsin. So if you think about it, all of these things are mixed together in the pancreatic juice. They get squirted into the duodenum. Trypsinogen gets converted to trypsin. This is now an active protease enzyme. It's an active enzyme, and it actually activates these two. Procarboxypeptidase and chymotrypsinogen get activated by trypsin. And by activated, these enzymes are proteins themselves, like this could be procarboxypeptidase, and trypsin comes along and clips off part of the chymotrypsinogen to activate it. All right, so that's what we're talking about. It's an enzyme that's going to clip part of those enzyme proteins to make them active. This is a diagram from your textbook, which is showing what I just talked about. So here are these. This is where the sphincter for that opens up and allows the fluids to come out of the hepatopancreatic ampulla. And one of those is this trypsinogen that you see here. And there are enzymes along the brush border that clip trypsinogen. It actually gets clipped by those enzymes to form trypsin. And then trypsin is an enzyme that takes chymotrypsinogen and converts it to chymotrypsin. It takes procarboxypeptidase and converts it to carboxypeptidase. And these are proteases that will digest proteins. Proteins coming in from your foods, along with the trypsin as well. And why are they doing that? Proteins are chains of amino acids. You can't absorb whole proteins very well. They have to be chopped up into individual amino acids before they can actually be taken in by the cells that line the small intestine and transferred over to the bloodstream. So that's why we need all of these important protease enzymes. All right, and one last topic before we go back to the digestion functions that are going on in the small intestine in more detail. What's regulating this? How does the gallbladder know when it's supposed to contract and squirt bile down into the small intestine? How do we know when we're supposed to produce more pancreatic juice and secrete that into the duodenum? So that'll be the topic of lecture 7. There are hormones, which we didn't really talk much about when we covered the endocrine system back at the beginning of the semester. There are hormones that control those functions, and so we'll take a little additional time out in lecture 7 to talk about those.